let's go ahead and take a second and go over kind of what you should have seen yesterday. And originally my intention was to have you guys narrate this. I'm going to put that on the back burner. That's not going to be a thing. So instead, let's go ahead and go through this together and make sure you're good. Now, that being said, I did post this on Schoology so that you can study from it, because it would be a good way to test yourself. Can you narrate it? Can you go through and describe what's happening throughout all the parts? And if you can't, that's a pretty good indication that you need a little bit more review of it. Also remember, as you're doing notes or as you're doing lectures, or especially when you don't have homework for the class, is a really good time to get a little bit of studying in. I get like a dog hair in my mask. Okay, 21 problems. So quick recap of mitosis and meiosis. Again, we've got mitosis starting out with a diploid cell. In this case, we're looking at something that is n equals 3. During S phase of interphase, all of the chromosomes will replicate. You'll end up with twice as much, but we know that this is in the form of chromatin, not actually chromosomes yet. But by the time we get to M phase and the start of mitosis, so the very beginning of prophase, we are going to have these sister chromatids visible, which are then going to get pulled onto the metaphase plate, separated, renucleated, I made up that word, pinched off, cytokinesed. We're going to go with that. And by the time we're done with mitosis, we've got 2 identical cells. Okay, so now here's really the difficult part. And I think people have a hard time with this. So here's what I want you to do. Hold up your fingers. Everybody, boop, 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 boop. Come on, at home. Boop, 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 boop. There you go. So here's what I want you to think. These two are sister chromatids, right? They're attached. They're on the same hand. These 2 are sister chromatids. These 2 are homologous to each other. When we look at the process of mitosis, what we're starting with is them just kind of, when they line up, they're going to line up like this, okay? What's important to remember about the entire process of meiosis is that instead of starting out like this, they're going to start out like this. Then they're going to separate the first time in meiosis 1, and then again in meiosis 2. I encourage you not to rip your fingers off just to demonstrate the separation of sister chromatids. That would hurt, and also you become a much less functional person. Moving on. All right, so let's go through meiosis and kind of keep that in mind. We've got the tetrads forming that separate during meiosis 1, and then the sister chromatids separate. So nothing different in interphase. DNA is replicated, nothing different when we get into prophase, but then we've got the tetrads forming. This part's a little weird to think of, but think of crossing over as like, you know, removing and resewing together your fingers on the wrong hands. Don't do that, actually. So crossing over is going to happen. Switch some genetic information. Liney, nice and random. Separate once. Renucleate. Da, da, da. 2 separate cells are now formed. We go into meiosis 2. The process happens again. And now this looks much more like mitosis, because you're going to have the sister chromatids that are getting split apart until eventually you make 4 nuclei and 4 cells. Okay. How is all of that? Is it starting to line up? Are we starting to see kind of some big pictures? Okay. So I'm not going to, ooh, hello, which button do I need? This is all good. Play in window, make it big. Okay. This is a good sign. So far, I still think everything's working technology-wise. All right. If you want to take notes on this, you may. You do already have the notes for mitosis versus meiosis. Okay. I just want to make sure we're super duper explicitly clear on those differences. So if you want to take notes on it, again, you can jot this down. But without further ado, quick kind of comparison of mitosis. Think of it like this. It happens from my head to mitosis. Okay, great. And when you think of meiosis, let me go ahead and write the word meiosis on the board. in a way that maybe will help you remember the function. You see this? I don't know if y'all can see this. Okay, look at the word meiosis. It literally has, can you see it online? Great, that's a sperm and an egg just chilling in the middle of the word. So what does meiosis do? Makes sperm and or egg, not and, just or. All right, so in mitosis, we know that we are starting with a diploid cell. The same thing is happening in meiosis. I feel like if anyone were to walk in right now, it would just look super sketchy. So I'm gonna get rid of that. Moving on. But the process is a lot different. In mitosis, we do prophase, metaphase, anaphase, telophase, and then we get into cytokinesis, which isn't actually part of mitosis, but just kind of bumps onto the back of it. Whereas in meiosis, we're going to have PMAT happen twice. We've got meiosis 1, so prophase, metaphase, anaphase, telophase 1, followed by 2. Okay. Here's the big difference. I don't know why I did not get this when I was in high school. I had the hardest time distinguishing these ones. Okay, let me be realistic. Probably even in freshman level college bio, okay? I don't know why it just didn't click. And had I realized the difference between the prophase in each, everything else would have made sense. So when that part clicked, it made sense. Hopefully this helps with you. So remember that in mitosis, the sister chromatids are together that are going to line up in metaphase, separate out, and then renucleate. Again, probably not really a word. Okay. Your result of this, 2 identical diploid cells. Exactly what you started with is what you're going to end with, unless there was like a weird little mutation that happened in S phase, but we're going to ignore that. Yay. Here's our difference. Whereas mitosis started with sister chromatids, meiosis is going to start with the homologs joining together. That's going to form your tetrads. That's when your crossing over happens. And therefore the tetrads are going to be what line up in metaphase, what break apart in anaphase, and what are then renucleated are the sister chromatids, plus or minus whatever crossing over occurred. It's then during meiosis 2 that those sister chromatids are together, they line up on the metaphase plate, are pulled apart, and then separated into 2 new nuclei. And by the time you are done, what we started with, right? We've got 4 pieces of genetic information, a pair of sister chromatids, a pair of sister chromatids that are homologous to each other. So they're going to separate once and once again, making 1, 2, 3, 4 cells, each one carrying 1 of each chromosome. All right. Mitosis is going to happen in your somatic cells. These are your body cells, resulting in somatic cells, or some germ cells, but very limited numbers. Your germ cells are the cells that are actually capable of meiosis. Those are chilling in your gonads. Gonads is a fun word to say. There was this really horrible video in like maybe the late 90s, early 2000s, and it was just called Gonads and Strife. And it just kept showing a squirrel in the video. It was a weird time to be alive, but anyway, that's what I think of with the word gonads. Moving on, germ cells. Germ cells are the precursor cells to sperm and the precursor cells to eggs. Females, you have a pretty limited supplies. You don't have any more germ cells that are getting made. You were born with whatever you'd have. Gentlemen, you're just gonna pump them out till you die. Yay. All right, the results of these are called gametes. The gametes are the haploid cells. And this is for both meiosis 1 and 2. That, I don't know if you can see that there is not a separation between the 2. Okay. Does that help clarify it all? Because I feel like I alluded to this a few times, but never really like clearly stated it all. Perfect. Yes, you're going to have 23 chromosomes in each of the human gametes and 46 in each of the germ cells and somatic cells. So all of our normal cells have 46. It's just going to be the gametes that are chilling with half that number. Okay. So to kind of be able, why did I do it this way? I think I missed a spot. I'll come back to this in a second. So super important idea behind meiosis is the introduction of genetic variability. That is going to be a big theme from now until the end of the year. Okay, evolution, really important to have genetic variability. Ecology, that's going to be really important. So for genetic variability, the ultimate source of genetic variation are the mutations that occur. Think of that as what's making each different card. What's making each different allele is mutation. Meiosis is all about shuffling those cards, bringing up new combinations. So we already saw that crossing over is going to lead to a ton of new combinations. Random alignment on the metaphase plate, 8.39 million different cells can be made from one set of germ cells or one germ cell. Okay. And then this piece here, random fertilization. That means that there's no way to necessarily know which of the gametes are going to be involved in fertilization. There's no rhyme or reason to it. Okay, so let's kind of contextualize, and this goes back to your point here. Here's the idea. We have a 2N cell, a diploid cell, that then doubles its genetic material. We don't call that 4N because it's just 2 copies of everything. So I like to refer to it as 2N, but times 2. So we double our genetic information. During mitosis, the result is 2N cells. When we start getting into meiosis instead, we're starting out the same way. 2N cell, replicating the DNA, making 2 cells that are kind of haploid-ish, with the exceptions of whatever crossing over occurred, to make 4 cells. Is that helpful? And now is baby making time, because when you introduce one of these cells at random, which one the lucky one is, just is a matter of chance, with another individual's haploid cell, when they come together, that is fertilization, and that is going to restore the diploid number back to 2N. 23 chromosomes from the sperm, 23 chromosomes from an egg, 46 chromosomes, if all goes according to plan. Okay? All right. Yeah, go ahead. Did I hear something?
So at that step, when the cell from mitosis meets the other cell, so do they combine the genetic information?
When the cells from meiosis do?
Yeah.
Yes. Yep.
Okay.
And so the result of that, what you guys saw yesterday when you were using the light and the dark colored, what those represent are the chromosomes that you would have gotten from each parent. In doing meiosis, you then made the gametes, which are really just a combination of both of your parents' genes. And that's just really a combination of your grandparents' genes. Starts getting weird as we look at it, but we'll continue from here, because we need to talk about your genetic makeup. Yes.
What part of
Say that again. Clarify that for me. Okay. So the reason that like different species are genetically isolated, I'm going to introduce that word. You'll hear it a lot with ecology. So the question was, why are different species not able to mix their genetic material? A lot of it comes down to, even if you mix them in a petri dish, the proteins from the sperm have to be very specific to the proteins that are on the eggs for them to even merge. Even if you were to take a really, really fine needle, which can be done, to take the genetic information from the sperm and inseminate it into the egg, even then, they won't combine, because if they have different chromosome numbers or their chromosomes don't look the same, then they can't start going through mitosis correctly. So it'll kind of fuse, but then when they go into mitosis, it's like, none of you are right. Ugh. And then they quit. And then the cells just die off. Emma. Huh? So what happens with a mule, they are hybrids. You have a horse and a donkey. They have different chromosome numbers. I think I'm totally making this up. I want to say 31 and 32, but I really don't know for sure. Or 30 and 32. Anyway, the chromosomes can come together well enough to continue to go through mitosis and grow an organism. But when it comes to actually making the sperm or the eggs, the meiosis process doesn't work because they don't have perfect homologs. So those don't come together. And so you can have a mule, but they're then not able to make the gametes, and therefore they're infertile. Oh my goodness, the questions. I'm terrified to ask Nick what his question is, but I'm going to do it. What, Nick? It would have to essentially have the exact chromosome makeup of a human, which the best way to do that is to use humans, which is why when humans and humans mate, they make humans, some of which are abominations, but I'm just kidding. I'm just kidding.
What about like monkeys? Did you mate?
Monkeys can mate with each other, yeah,
We're kind of like.
but we're not. Our chromosomes are different enough that they are, I'll say this, when experiments have happened, they have not been successful. Now, there have not been a ton of experiments because of some obvious ethical
When you're
concerns.
saying.
The question right now for you guys who didn't get the luxury of hearing that question was, can humans make baby human monkey hybrid things with monkeys? I'm trying my best to be tactful. It's not going well. Okay, what was your question?
What makes people infertile?
Ooh, super good question. What makes people infertile? A lot of different things can be involved. So it can have to do with not having the right hormones that cause the development of either the sperm or the egg. So in males, you have testosterone that leads to the development of the sperm and having their tails and everything. If the balance of that isn't correct and they don't develop properly, they're not going to be motile. They're not going to be able to swim. For females, there's a ton of issues that can happen with estrogen, progesterone, follicle stimulating hormone that cause, remember I said you start with all the eggs that you're ever going to have. So that means that really they're kind of arrested partway through meiosis 1. So females don't actually have mature, ready to, like, they're not done meiosis until after the sperm has entered into the cell. Like, they just kind of quit and kind of freeze in place. And so you have to have just the right hormones to stimulate those cells to then continue through meiosis to get into part of meiosis 2. If that doesn't happen, you don't have viable egg cells. They can be released poorly. There can be inadequate tissue in the lining of the uterus that won't support the baby. So many things can go wrong. The fact that you're here is kind of a miracle. Like, consider the fact that males make how many thousands of sperm per day, but it just so happened that to make you, the correct sperm to make you happened at the same timing of your, not to be weird, but your mom's ovulatory cycles, and you managed to survive. Like, it's estimated that Oh, no. You said, what a shame. It's really, okay. Estimated, oh, it's estimated that something like 50 to 70% of fertilized eggs, sperm, egg, life, never embed into the uterus, and so just get quite literally flushed down the toilet or washed away. So that's crazy. Yeah. Yes. Yes, that's a cell signaling type thing. Actually, this is really cool. As soon as fertilization happens, a cell signaling cascade results that causes a massive dump of chemicals from inside the egg cell that produces like a really hard shell around the egg so that no other sperm can like get in there. It's like, no, no, no. I found my winner. Everybody else stop. And then fertilization can't reoccur, but there's some weird stuff with chimeras that maybe, I don't know, magic. Yes. How do you have what? That would be the sperm, the egg, they fuse, mitosis, 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 mitosis. And then instead of going mitosis, mitosis, mitosis and staying together, two clumps of cells break apart. And then they just continue to develop normally. So that happens really, really early before they've even made that, like, remember how we kind of talked about the hollow ball of cells and like the little cells on the inside that become the baby and then the placenta? Based on like ultrasounds, you can determine exactly when they split, because if identical twins share a placenta, then that means they split after those cells kind of differentiated from themselves, but before the inside cells started really turning into a baby. And so you can have identical twins that share a placenta or that each have their own placenta. And then you can have fraternal twins, which is when two eggs are released, both fertilized, both implant, both grow, and then both sleep at completely opposite times of the day for 3 years, and make mothers slightly crazy. Now you understand my problem. Yes. A ton of DNA replication and cell signaling to start indicating, hey, you're going to be a foot, you're going to be a brain. Separate based on that. Foot cells like, I'm a foot, I'm a foot, I'm a foot. Then the cells near it are like, okay, it'll be a leg, this is great. And so forth with the brain. It doesn't happen at all like that. Like, it's very, very metaphorical. Oh man.
Hunger.
Yes.
Why is ovulation a time where women are most likely to get pregnant?
Sorry, I'm having like a hard time hearing you very well. So the whole process of, oh man, is it okay that are you guys terribly upset that we're off topic? Is anyone like crying about this right now? I love this stuff if you couldn't tell. So what has to happen during a cycle is ovulation. Y'all know the 28 days. First of all, do you know that a woman can't be 1 or 2 weeks pregnant? Did you know that? They count pregnancy as 40 weeks from the date of the start of the menstrual period before she got pregnant. So someone who is just pregnant, like it just weaseled its way into that uterus, is considered 3.5 weeks pregnant. Because usually it's during the 3rd week that they fertilize, and then a few days before it can implant. Anyway, that was an aside. So what has to happen is the egg has to be released from the ovaries at just the right time, that the semen is almost at that, like the end of the fallopian tube. At that point, they meet, fertilization happens, and then that little ball of cells is going to continue to grow and grow and grow. And so it's got to be right after the egg is released that that fertilization occurs, because otherwise the egg's either going to be still in the ovary and therefore not going to be privy to the sperm cells that are around, or it's going to get all the way washed out, in which case that's not beneficial either. Yeah, Emma. You don't eat twins. You can't eat your twin in the womb. What can happen, and this is one of the big dangers, like especially as a twin mom, I had a lot of other people in my network that were twin moms, and anyone who has identicals, especially if they share a placenta, the worry isn't one of them being like, nom, nom, nom. The question's about eating twins, sorry. It helps if I like clarify this. The umbilical cords, each attached to the uterus, if one of them has, or to the placenta, if one of them has a smaller umbilical cord, then the one that's growing better will continue to establish a stronger blood flow to them and can basically starve the other one. Usually it's in the first few weeks that, you know, once after, like right after a female has found out she's pregnant with twin, well, pregnant at all, it would happen. So most of the time, if that happens, it's undetected. It really only is known about when, for some reason, someone finds out that they're pregnant really early on. Yeah, Sarah. Yes. I might as well. All right, this is going to be a later topic, but we're going to talk Down syndrome for just a moment. Okay. Am I in meiosis right now? Or mitosis? Yeah. So what happens in cases of what are called trisomies, getting 3 of one chromosome is called a trisomy. Whereas like a triploidy would mean 3 sets of all the chromosomes. So haploid is 1 set of chromosomes, diploid is 2 sets of all chromosomes. We don't end up with a triploidy, we end up with just a trisomy, getting 1 extra chromosome, and that's going to happen as follows. Remember how I said that females have their eggs kind of arrested during meiosis. So they're going to look something like this. Okay. Then each month, the chemical signals come in to continue differentiating. One of these is going to be selected as the one that develops. The other is just going to be a supporting cell. It's not really going to actually become a gamete. Is it considered a gamete? I would have to check that, but it doesn't become what can be fertilized. Okay. So then at this point, what we're doing is getting ready to go into prophase 2. If instead of they separate, right? Let me play in window. They should go but if instead they go what you're going to end up with is one of the gametes will have 2 copies of one of the chromosomes, which when it then gets fertilized and gets another copy of the chromosome from the sperm cell, makes 3 copies of that particular chromosome. We cannot survive with a monosomy except for the sex chromosomes, but we'll talk about that later. So this would basically not be a viable for, pregnancy if it were to get fertilized. Put that guy back. Oh, Naomi. Okay, identical or single, would we call it an embryo? Whatever. Single little ball of cells, split, 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 and not quite. Then developing continues. So you can have conjoined twins from anywhere along that part of the body that the 2 cells were still conjoined. That was super redundant. And Ashley, what was your question? Depends, and it can be a combination. So usually there's been a couple documented, okay, so identical triplets are super rare. Most of the time what triplets are either 3 separate eggs that are fertilized or 2, and then one of them splits to identicals. How much would that suck to be the 3rd one? Like, oh, these are my identical twin siblings, but I'm like just ugly. I don't know where that came from. But yeah, so triplets and higher order multiples tend to be from some combination. Once you get into twin, I think there's only been 1 or 2 reported cases of quintuplets that survived to birth that were 2 sets of identicals. Most of the time, then it's just a matter of in vitro fertilization, which is take all the eggs out, mixy in a petri dish, shove as many as you can back into the uterus and hope something sticks. Sometimes they go a little bit crazy and they're like, let's increase our chances and put 14 in there. And then you end up with octuplets. John and Kate plus 8, the reason that they have 8 kids is the first 2 were twins. The IVF clinic said, what do you want to do with the rest of these embryos that we have? And they were like, well, we can't really kill them because that seems mean. And we don't want to donate them for research because that's going to kill them. And that seems, let's just pop them in, see what happens. Sextuplets later. Oh my God, I can't imagine. My brain hurts from the lack of sleep that would ensue. Emma, what was? Yeah, the first 2 and then the next 6. Yep. Oh, yes. They were all fertilized at the same time, which means 2 of, oh. So I'm going to tell you a secret that even my children don't know. They don't know who's older. But because of how it is quite possible that the one that is further up, and there tends to be the younger one in terms of birth order, is the first one fertilized, which is why they're higher up. So my son is kind of older than my daughter because he was probably, assembled first. But since they removed the girl child first, she's technically older, but she doesn't know that. And we're not going to tell them that because we don't want them to have a weird complex. So instead, they just think they were hatched. Yeah, Sarah. Yes. Yes and no. The question was about IVF. Do you have to artificially induce them to get into meiosis 2? And what they actually do is they get the female to hyperovulate by giving her a ton of shots that have those chemicals. That forces her into meiosis 2, which then, when it's introduced to the sperm, continues the process. And then they put them back in. Gabe. All of them. All of them at once. We're just like, here you go, Kate. Have some babies. It's super unethical. Super unethical to do that. Because of, well, the other option is just putting a few back in a time. And if that doesn't work, then you have extra embryos. But they were just like, let's put, I think they put like 12 or 14 in simultaneously. And usually they don't do more than 3. So.
Miss Unger,
Wasn't looking so good. Yes.
is it possible for like a malfunction to happen and the mitosis 2 doesn't go through with it and you just stop at meiosis 1?
Probably. The question was, is it possible that the meiosis 2 process just never completes? Yeah, it's probably pretty possible and more common than we know about. But if fertilization occurs and implantation occurs, there's not going to be any development. So it's either going to be absorbed by the mother's body or menstruated out. Menstruated. That's a weird verb. Is it a verb? I don't know. This is not going to make it back onto YouTube. Don't worry. Okay, Ashley. The question was, is IVF more likely to have issues? Can more things go wrong because it's artificial? And that's a loaded question because the people who tend to have IVF can't get pregnant naturally. So it becomes a question of, if there's issues, is that a function of the mother's biology or is that a function of problems with the IVF? Usually it's advanced maternal age, which, going back to trisomies, the reason that women over 35 are so much more likely to have a child with Down syndrome is because those eggs have just been chilling there for 35 years. Whereas younger women tend to have fresher eggs. I'm sorry if I'm making these people sound like chickens. Emma. And I know someone online had a question too, and I'll get right back to you after that. Emma. Identical twins, one has Down syndrome. I'm hesitant to, I can tell you it's probably incredibly uncommon if it happens at all. My thought might be that early mitosis, there could be a non-disjunction occur. So when it separates, one gets an extra set of the chromosomes. And then the other one would, that lineage would have to die off. I don't know. Huh. Were there a set of twins here that was the case? I mean, okay, because if they're identical, yeah. Huh. I'd have to look more into that because I'm sure that there's something, but it wouldn't be an issue with meiosis. Fun story, there's also a way to have identical twins where one's a male and one's a female. Yeah. Any idea on how that happens? Magic. Right after they start to separate or to develop, there's a random mutation in the SRY gene of one of the males. So they're both genetically male, but then the gene that says be a dude mutates randomly in one of them. So that becomes, that embryo starts developing as a female, even though genetically they're identical with the exception of a couple mutations here and there. Ida, was it you that had a question, dear?
No, I think it was
It was me.
Go ahead, Anna.
I actually have two questions now.
Okay.
My first one is like, so the meiosis we're studying now, is that just for like males?
It would be pretty much the same in males and females. The difference is that it just kind of stops at certain points for females, right? So we stop right about here-ish, but then we keep going once ovulation or once like the hormone signals are coming in and once fertilization occurs. In males, they're just like all the time, all day, every day, tons of them.
And
Okay.
then like,
oh, sorry. The question was, is it the same for males and females? I hope you heard my answer. Okay, sorry. Anna, go ahead.
and then like, how are like they're identical twins if like all the cells that go through meiosis end up like different?
Okay, so how can they be identical if all the cells that go through meiosis are different? It's because they started as just one of those gametes from the male and one of those gametes from the female. So they start out as one cell. So that cell, as it's mitosing, eventually splits to make one clump of mitosing cells and another clump, but they're already diploid. They're back to that 2N stage at that point. Game. And then like one more question after this, and we will continue, but I like this. The question was, the question was, is it possible to remove a growing embryo and then reimplant it? My inclination would be to say no. Because you would need not just the embryo, but you need to get the placenta. And once the placenta is formed, I don't think you would be able to implant it. Like, I don't think it would take from the mother because it has a different genetic identity from the mother. So my guess would be she'd try to kill it because of the immune system.
How do you?
They're identical.
Oof, the questions are getting good. I just thought of something crazy. It would be possible to clone someone, to take cells that have developed into embryos in a petri dish, right? Split them, have two identicals, implant one, freeze the other for 20 some odd years, and have a mother have her own identical twin.
I love it. I love
He loves it. He just wants everyone to know. Okay, I am
Maybe
going
I
to.
want make that my life work. Making a woman have herself.
What is happening? I don't know. I don't know. Everyone that's online is probably just like, oh my God, thank God I can turn on Netflix right now.
Like a woman having the baby for you.
Yes, surrogacy.
So how is that not work?
Because with surrogacy, what happens is it's the embryo itself in the very, very earliest stages. So you could have an embryo, you could split it apart and then continue to allow it to grow in vitro and then implant them into two separate women, the same woman. And if they both stick, they'd be identical twins, but potentially different carriers. Like what do you call a surrogate mother? I don't know the proper like kind term. And I feel like a womb for rent is inappropriate. Okay, go ahead. It would be just multiple different sperm, each fertilizing their own egg. And so that's fraternal twinning. My brain was going somewhere, then it didn't. Random fun story, certain organisms, kangaroos, I think there's a few other ones, actually have split uteri, split vaginas. There we go. That's what they have. And then it leads to two separate uteri. And so what happens is it prevents two embryos from both growing in the same area. So it prevents that fetal eating twin syndrome. But that also means that the males have to have forked penises so that it can fertilize each side, each horn of the uterus.
Wait, kangaroos have
Forked.
oh.
No. Kangaroos do not have well, no, they don't. Okay, let's talk about genotype and phenotype because that was off the rails on the crazy train, and that's fine. Genotype, phenotype, just to make sure we remember some of these terms from earlier. Here I was thinking this was about to be the fun part. So genotype is referring to the genetic makeup. We're going to introduce two new words that if you've not seen yet, you'll want to make sure to know them: homozygous and heterozygous. So homo meaning same, zygous meaning of genetic material, zygote. So a homozygous trait is one that you have two of the same alleles for, whereas a heterozygous trait, you have two different alleles for that trait, okay? If we take a look back at at you guys, at your chromosomes from yesterday, my guess is that you've got a pretty good smattering of homozygous and heterozygous traits when you go to start. The phenotype, and this is where a lot of people get confused. The phenotype is how those genes are expressed. And different types of alleles have different types of interactions, but we're going to go over the main type that Mendel came up with. So the phenotype is going to be determined by which alleles are present and how they interact. Okay, so what I want you guys to do is determine what you look like. So as it turns out, dragons have an N number of 3. They have 3 chromosomes. Chromosomes 1 and 2 have genes A through whatever it came up to. And then the sex chromosome had KL, HIJKL, yeah, K and L and M. So you're going to figure out what your genotype and phenotype are for your dragons. So to be able to do this, you have a lovely this thing, one of gotta publish it, which I'll do real fast. Okay. So you are about to find out what you look like. We might not get to have sex today. I'm just putting it out there, but it's okay. And so we learned that eggs can really stay good for quite a while. So if they have to stay another day, that'll be fine. Oh my gosh, I cannot get the computer to do anything. All right, so if you go to the folder in Schoology that says patterns of inheritance, there is a table there that have the genotypes and phenotypes. Now I want to take note of something. These are going to have a lot of complex interactions. So for right now, you just need to take note of what your genotype and phenotypes are. So for example, if you take a look at your chromosomes and you have 2 big A alleles for the A gene, that means you have the homozygous yellow eye trait. You have big A and big A. And in this, if you use the table, that would indicate then yellow whites of the eyes. I didn't know how to say that without being like, it's Clara. Y'all don't know. Anyway, maybe you do. But the whites of the eyes would be yellow if you are either big A, or big A, little a. Only if you are little a, would you have white eyes. So you can use this chart to determine what your whites of the eyes are, your scaliness, the shape of your spikes, your claws, your tail, your iris, and your wings. A couple of things get a little bit wonky down here with fire breathing and teeth. If you are female, that means you have 2 copies of the chromosome that had L and K on it. So you're going to be reading the top row. If you're a male, you only get one of those genes, which means you're either big K or little k, no heterozygous option there. For skin color, you're going to need to look at the D, F, and J genes to determine how many of the big alleles do you have, and that's going to determine your overall color. Okay, so what I want to do is give you guys about 10 minutes to go through and write down what are your genotypes and phenotypes for each of these traits that are here. Yeah, Nick, go ahead. Did you just have a hand up? I saw it. Grace, did you put a hand up? I saw. I'm hallucinating. All right. You can honestly, if you want to put it into your onto the keynote that you did, you can do that. So long as you know where it is and you can determine what your dragon, sorry, what you, my little dragon friends, all look like.
So do we just like choose what we want from the chart?
Nope, choose based on what your chromosomes were, based on the coin flipping from yesterday. So for example, one of these should be what I need. No, that's not what I need. Ah. Yes, where it says the original genotype. I could have just said that and that would have been much smarter. Not very smart though. I can't get it up, which is super fun. There we go. So on the slide that said your original genotype, you'll have either, whatever color you have, but you changed some of the letters to lowercase. So for example, for the purple one, if you have them next to each other, your genotype is whatever letter this has on the dark colored chromosome and the light colored chromosome, and so on and so on. If you have little letter, big letter, the way you would write that would just still be big little. So if you are heterozygous, if you have one of each, still write it as the capital first and the lowercase second. Ashley. They're at the bottom. They're all working together to determine skin type, skin color.